Hello students, today we will take up the third lecture on plant hormones and we will discuss ethylene and abscisic acid in this lecture. Let us start with the introduction of ethylene. Unlike the rest of the plant hormones, ethylene is a gaseous plant hormone and this group of plant hormone is represented by ethylene only. Ethylene with the molecular formula C2H4 is a tiny gaseous molecule and has the simplest structure among all the non-plant growth substances. The first study which contributed to the discovery of ethylene as plant hormone was by Dmitry Nelzhubov in 1901. Who observed that ethylene from illuminating gas produced a peculiar growth consisting of a shortened and thickened epicotyl and horizontal banding in etiolated pea seedlings. Later, in 1917, Dowd discovered that ethylene stimulated abscission and in 1934, Richard Gane discovered that plants synthesize ethylene and was the first gaseous signaling molecule to be identified in any organism. Ethylene is produced essentially from all parts of higher plants and affects many physiological aspects of plant growth, development and survival, and it also mediates many adaptive responses to both biotic and abiotic stresses. However, ethylene is best known for its role in climacteric fruit ripening and the triple response. Ethylene is commonly synthesized at or near its site of action. Being a gaseous hormone, it moves within the plant by diffusing across membranes into nearby cells and does not require any transporter protein for its delivery to target cells. Let us now see the biochemistry of ethylene. Ethylene is a simple hydrocarbon having the formula C2H4. It is commonly produced in most tissues of higher plants from methionine, which is converted to S-adenosyl methionine or SAM. S-adenosyl methionine is then converted to 1-amino cyclopropane 1-carboxylic acid or ACC, catalyzed by 1-amino cyclopropane 1-carboxylic acid synthase or ACC synthase in short. And finally, ACC is oxidized to produce ethylene catalyzed by an oxidative enzyme called ethylene forming enzyme. Ethereal or ethiphon is 2-chloroethyl phosphonic acid and is an ethylene derivative sold commercially, and when applied to plants in solution form, these compounds release ethylene which produces its effects in plants. Let us now see the physiological actions of ethylene. Ethylene affects many plant processes and the main effects of ethylene are: first, ethylene influences germinating seedlings producing a set of responses termed as triple response, characterized by the thickening and shortening of hypocotyl with pronounced apical hook growing horizontally without responding to gravitropism. The second effect is ethylene stimulates the release of dormancy and induces seed germination. Thirdly, ethylene promotes leaf and flower senescence. It also stimulates leaf and fruit abscission. The fourth effect is ethylene promotes ripening of fruits and induces an increase in respiration in some climacteric fruits, causing additional release of ethylene. Next, ethylene plays a role in induction of femaleness in dioecious flowers. The sixth effect is ethylene influences geotropic responses, as ethylene produced by higher concentration of oxygen reverses the geotropic curvatures caused by oxygen, leading the roots to curl upwards instead of growing downwards into the soil. Next, let us take up the mode of action of ethylene. The first mode of action is the enzyme induction by ethylene. Ethylene has been reported to induce a number of enzymes including acid phosphatase, ATPase, alpha amylase, cellulase, chitinase, chlorophyllase, cinnamic acid 4-hydroxylase, cytochrome C reductase, diaphorase, B1,3-glucanase, invertase, malic enzyme, pectin esterase, peroxidase, phenylalanine ammonia lyase, polygalacturonase, polyphenyl oxidase, protease and pyruvic carboxylase, etc. by reducing the lag time or increasing the rate of enzyme activity. The next is the effect of ethylene on RNA synthesis. In some fruits like apples, ethylene has been shown to increase RNA synthesis followed by an increase in protein synthesis. However, the increase in RNA synthesis could not be observed in pre-climacteric bananas or avocados treated with ethylene. The third mode is the effects of ethylene on chromatin activity. Ethylene has been shown to regulate RNA synthesis, and the mechanism involves change in chromatins of ethylene-treated tissues, leading to change in quantity and kind of RNA. The next is the effects of ethylene on DNA metabolism. The growth of different plant tissues is either promoted, as in case of swelling, epinasty, hook closure, seedling elongation and seed germination, inhibited, as in case in arrested development of buds, leaves or apical meristems, or unaffected, as in case of mature tissues such as stems and leaves which do not undergo any change in response to ethylene. By ethylene may be mediated by regulation of DNA synthesis by ethylene through its action on microtubule formation during mitosis. Next is the ethylene action during fruit ripening. Fruit ripening process involves rapid increase in respiratory activity, and as the rate of respiration increases, ethylene exerts its effect by affecting membrane permeability and activating genes. The activated genes results in the synthesis of specific mRNAs, which are translated to specific proteins such as pectinase and cellulases that act on the middle wall and primary walls, loosening the cells, which converts the hard fruit into soft fruit during ripening. On the other hand, the effect of ethylene on membrane stability and permeability results in leaking out of pigments from the tonoplast. Further, ethylene induces the degradation of chlorophyll and the synthesis of some anthocyanins, which give attractive coloration to the skin and the flesh of the fruit. Thus, ethylene helps in turning the mature fruit into a softer, sweeter and colorful one during ripening. Next, let us see the ethylene signal transduction pathways. Ethylene signal transduction pathway generally involves the permeation of membrane by ethylene followed by binding to a receptor on the endoplasmic reticulum. The receptor releases the repressed ethylene insensitive protein 2 or EIN2 in short, which then activates a signal transduction pathway leading to activation of regulatory genes that eventually trigger the ethylene response. Ethylene-induced gene expression is mediated through a cascade of kinase activity, and the activated genes code for an enzyme used for ethylene biosynthesis. Let us now learn about the next group of plant hormones, that is abscisic acid. Abscisic acid is represented by a single compound like ethylene, unlike auxins, gibberellins and cytokinins group of hormones. Abscisic acid discovery first started with the identification and characterization of two compounds called abscicin 1 and abscicin 2, responsible for the abscission of fruits in cotton by Frederick Endicott and his associates in 1963. During the same year, two other groups led by Van Stevenink and Philip Waring, studying abscission of flowers and fruits from lupin and bud dormancy in woody plants respectively, discovered a compound similar to abscicin 2 and named it as dormin. Abscicin 2 or dormin is presently called abscisic acid or ABA in short. It is an unfortunate name since subsequent research has shown that ethylene rather than abscisic acid controls abscission. Next, let us learn about the biochemistry of abscisic acid. Abscisic acid is synthesized from carotenoids in plants, mostly angiosperms and gymnosperms, but not in liverworts, and synthetic abscisic acids have not been reported. Abscisic acid is a sesquiterpene with molecular formula C15H20O4 and its molecule contains a carboxyl group, 4 double bonds and 4 methyl groups. Abscisic acid is synthesized as an inactive form which is later converted to the active form. Unlike the polar transport of auxins, it is transported non-polarly in both up and down direction in xylem, phloem and parenchyma like the gibberellins and cytokinins. Now let us see the physiological actions of abscisic acid. Abscisic acid affects a number of plant responses in tissues of all ages. The main actions of abscisic acid are: first, abscisic acid acts as an abscission accelerating hormone in many fruits and leaves. Secondly, it induces dormancy in some woody plants. Thirdly, abscisic acid inhibits shoot growth but promotes the growth of roots. Next, it stimulates the closure of stomata in response to water stress. The fifth effect is it induces the synthesis of storage proteins in seeds. Next, it also inhibits the effect of gibberellins on stimulating the synthesis of alpha amylase. Finally, abscisic acid is also thought to play a role in plant defense by inducing transcription of proteinase inhibitor genes in response to wounding. Next, let us learn about the mode of action of abscisic acid. First, let us see the role of abscisic acid in stomatal closure. The mechanism of action of abscisic acid or ABA in stimulating the closure of stomata during water stress is not clearly understood, though is commonly believed to involve the regulation of membrane permeability. The next is the role of ABA in dormancy. ABA serves as a potent inhibitor of seed germination or the maintainer of seed dormancy in many genera of higher plants, and the effect is exerted in a reversible way in which the inhibitory effect can be removed and the seeds can resume growth when ABA concentration decreases. Next is the effects of ABA on abscission. ABA is not closely involved in the regulation of leaf abscission, and exogenously applied ABA has been shown to have little effect on leaf abscission. However, ABA has been reported to be involved in acceleration of fruits abscission. The next mode is the role of ABA in wilting. The level of abscisic acid or ABA generally increases in plants during wilting, and it leads to reduced growth of plants during drought. The extra increase in level of ABA is brought about by synthesis rather than release from a precursor. The next one is the action of ABA on nucleic acid synthesis and enzyme activity. ABA has been shown to have little or no direct effect on the synthesis of nucleic acids and the activity of enzymes. The next is the involvement of abscisic acid in messenger RNA translation. The mode of action of inhibition of activities of various enzymes like protease and isocitrate lyase during the germination of seeds by ABA involving translation inhibition of pre-existing messenger RNA in the embryo has been shown to be the effect of ABA on inhibition of RNA synthesis. Coming to the conclusion. So, in conclusion, we see that a number of physiological processes of plant growth and development are either promoted, as in swelling, epinasty, hook closure, seedling elongation and seed germination, or inhibited, as in arrested development of buds, leaves or apical meristems by ethylene depending upon the tissues involved. As a result, there are many mechanisms of ethylene action, as there are different modes of ethylene operation. Even though ethylene affects a number of physiological and biochemical processes in plants, much more work is still required to discover the mechanism of action of these processes. On the other hand, although ABA was first discovered in dormant leaves, this hormone seems to have little or no effect on abscission of leaves or dormancy of buds. However, ABA appears to play some role in fruit abscission and stomatal closure in response to drought conditions. The mechanism or mode of action of ABA is also not fully understood and needs further research. Thank you.